Welcome to a CUNY TV virtual roundtable discussion, COVID-19, a crisis in motion. I'm Mike Gilliam. Well, we've reached a grim milestone in our country. More than 200,000 Americans have died from the coronavirus. That's more than any other country on earth, and many believe the actual number of deaths is even higher. To put that in perspective, that's more than the number of people who died in World War I, the Korean War, Vietnam, Afghanistan, and Iraq combined. It's enough to fill Yankee Stadium about 4 times, or the total population of a city like Rochester or Salt Lake City or Tallahassee, Florida, or about 70 times the number of people killed on 9/11. Right now, the majority of Americans are bracing for a devastating second wave of the virus and pinning their hopes on the development of a vaccine. No vaccine exists, but people at the City University of New York are doing their part to find one and to also aid in the battle to survive the pandemic. There is a lot going on across CUNY, from research into vaccines to studies designed to help locate where coronavirus is lurking and being spread without hands-on testing of individuals. We have a number of people to talk about their work, including Queensborough Community College Associate Professor of Biology, Monica Trujillo. Angelique Corthals is a researcher and associate professor at John Jay College who's been studying bats and COVID-19. A member of the CUNY Coronavirus Task Force, who is a professor of health and policy and management at the CUNY Graduate School of Public Health and a contributor to Forbes magazine, Bruce Y. Lee, MD, and Paul Gottlieb, a microbiology professor from the CUNY School of Medicine, who's also a researcher and scientist. Dr. Gottlieb, let's start with you. Working with the company Technovax, you had a breakthrough in novel coronavirus vaccine research. Tell us about it.
Well, we have assembled something called the virus-like particle in mammalian cells. And this amounts to the outer shell of the virus, absent genetic material. And of course, the idea behind this is to use it as a vaccine. It can't replicate, it can't cause illness, and yet due to its surface structure, it has all the proper immunogenic determinants that should be recognized in a proper vaccine. You can almost imagine it as one of the classic inactivated vaccines, such as poliovirus, except this is made by recombinant means. Again, all research, not all, but most research these days is quite expensive. It's becoming big science, and it demands collaborative research. And so we are working with, as you mentioned, the Technovax Corporation of Elmsford, New York, with a very fine vaccinologist originally from Wyeth Laboratories, Jose Galarza. The intellectual property is developed by Technovax, and they are assembling these structures in mammalian cells in the hope that much of it can be made, because you have to make an awful lot if you're going to vaccinate everyone on Earth. And we are doing the structural work to confirm the actual positioning and density of the outer shell immunogenic determinants. We're collaborating with a very bright fellow here at City College, Reza Hyatt, originally of the Scripps Institute, who has engineered the surface proteins so they're in a special conformation. We can change some amino acids that did this computationally. And therefore, we hope it's going to be very immunogenic for 2 reasons. First, because it's presented as a virus structurally. You might have seen that in the press release images. And second, because the forced conformation of the outer proteins are considered to be highly immunogenic. So we're very, very encouraged. We intend to start animal studies as soon as possible, which is always the first step. And so we consider this a significant breakthrough and a significant collaboration with our public university and a private corporation.
So how did CUNY come to be involved?
Well, I've been working in collaboration with the Technovax Corporation for almost 10 years we've worked on many vaccines focusing on respiratory vaccines, adenovirus, respiratory syncytial virus. In fact, we even, it's not respiratory, but we also had a prototype for a Zika virus vaccine when that was an issue. So we've been putting a lot of effort into this, and there's a phrase that we use now, it might change in the future, called vaccine platforms. And the overall scheme for us is that our platform is the assembly of these types of particles that are all related because of their assembly in mammalian cells, which, for lack of a better phrase, makes them similar to what you would expect to see in the natural world.
Bruce, it's not surprising that CUNY is in fact involved in trying to come up with a vaccine, right?
Yes, certainly. I mean, there's a lot of expertise within CUNY across many areas of vaccines, so it's not surprising.
Mm-hmm. Okay. Angelique, talk to us a little bit about your research. We usually think of John Jay as a place where law enforcement is studied, but you've been studying bats, right?
Correct. Yes, we do have a science department. So yes, my work is actually focused on two things: the immune system, and particularly the relationship between metabolism and immune system, and I also come from the pathology worlds. So I have a pretty good understanding of anatomical features that may be implicated in SARS-CoV-2. And this is why I partnered with my spouse, Dr. Liliana Davalos, at the Stony Brook University, the Department of Ecology and Evolution, who studies bats for a living. And we combine our two areas of knowledge to look into why bats have this notoriety for being resistant to viruses, but they're in fact not resistant to all viruses. They actually do get sick from some of the viruses. However, coronaviruses seems to not affect them as much as they affect us. And so we're looking at, with Texas Tech University, we're looking at several species of bats, and we're looking at all of their entire genomes, particularly focusing on their immune genes. And with Yale University, we are looking at the nasal passage of bats, which is something that most people haven't been looking at. It's in fact a completely groundbreaking research that Liliana's doctoral student, Laurel Yall, who's now a postdoc at Yale, started doing in relationship to the olfactory senses of bats. And as you know, or may know, bats, some bats have that extra organ, and that extra organ is called the vomeronasal organ, which sort of stands. It's an extra organ to sense smell at the back of your nasal passage, almost close to the palate, which multiplies your sense of smell by thousands of more molecules and much smaller molecules. Now, why is that important in SARS-CoV? Well, because some bats have it, some bats don't have it. And the bats that don't have it actually have a nasal passage that's very similar to humans, and their epithelium is also very similar to humans, but they don't seem affected by coronaviruses. So what is it about this epithelium that sort of may be receiving the virus, but doesn't get affected or doesn't get the individual bat infected? So the bat that I'm talking about is called Rhinolophus Cynicis, which is a bat that was actually implicated in the SARS-CoV spillover. We're not entirely certain that that's the case. We know that there is a SARS-CoV-2-like very close ancestor that's been circulating in those bats for at least between 70 and 40 years, so it's had time to ramp up into that population. The actual same SARS-CoV-2 has not yet been found in those bats, but it's very likely that the spillover came from that reservoir. And so the idea of our research is to combine that sort of anatomical part of the bats and the genomics part of the immune system of those bats, and find out what makes them so much more resistant, or at least resistant to those viruses that can infer points of therapy for SARS-CoV-2 in human, and can also give us some extra epitopes to look at for vaccines.
So Angelique, for a layperson like myself, take us inside the lab and just kind of describe for us the work you're doing with these bats. What's it look like?
Right, so we don't have a colony of bats in the lab, just so that you're aware of that. Most of the work is actually done on computer, because we do have the assembled genomes inside the lab, and all we have to do is literally hours of computing and making sure that we are aligning each of the genomes, etc., etc. Then we have to do a little bit more than that. We actually have to look at tissue samples that we've actually already collected in the wild. That's why we were so it was so interesting for the NSF to fund us, because we already have the material and perform histology on them to look at the actual anatomical difference between the different bats.
Okay, great. Monica, your turn. Let's talk a little bit about your research. Now, it's a little bit different than the others in that you're not aiming to create a vaccine, but instead yours involves testing, the testing of wastewater. Kind of describe it for us and tell us what you're up to.
Yeah, I think that I would like to start by saying that the way this project started, I think it talks about the power of CUNY. So I was teaching when the pandemic hit, and I was teaching a general microbiology class. I had a research component that we were analyzing the soil microbes in Newtown Creek. And when the crisis forced us to go online, I said, what am I going to do with my students now? And so I decided that the only thing we were going to do was to read papers about COVID-19. So I sent them, I had to read them all, right? And I had to try to kind of decipher it to them. These are community college students. Just to give you a little bit of background, I teach, mostly to nursing students, but this microbiology class are students that want to major in biology. Most of them are looking into transferring to 4 years, and they want to have some research experience. So across all the papers that I read, I came across a paper published by a group in the Netherlands who very early on were able to detect the presence of the virus in wastewater in the airport, right? A couple of days after the first case was identified, they saw the signal. So my connection was pretty fast, thinking, can we do this in New York?
Let's get to that portion of your research now.
Yeah.
So basically, you wanted to test wastewater.
Yes.
Why, and what did you hope to find out?
We wanted to know if we could detect the genetic signal of SARS-CoV-2. The value of that is that you have like a measurement in each one of the wastewater plants. Each one serves different parts of the city. So if you know how many molecules, how many copies of the genome of SARS-CoV-2 you have, that gives you an idea of how many people are infected.
So this is better than actually just going out and testing those who may have already been deemed to have symptoms, correct?
Yes, because these you get like a big bag in which everybody who's shedding the virus, it's going to be counted.
And so then you have a better sample and you can tell exactly what's going on in a particular area.
Yes, because you add to that, you add to the value of grabbing everybody who's infected, you overlap the geographical distribution.
How's it gone so far?
It's going very well. We feel very proud that the New York City DEP has adopted the CUNY protocol, and that in their hands, it's reproducible and it's being used for surveillance.
Okay, I kind of want to go back to some of our other guests now and talk a little bit about some of the other research that they're doing, and also kind of get their reaction to exactly what each of you is doing at this point. Paul, when you listen to your colleagues here, Angelique and Monica, what's your impression of this type of work that's going on?
Well, I think it's very interesting, the bat story, and I think it should be emphasized that although there's a lot of false information, it certainly appears that this virus has been a zoonotic transfer from bats. And if you look at the genetic sequence, I think, Angelique, you would agree that there's very little change, just enough to adapt it to the human transmission. And I think that has to be emphasized and explored even further. There actually was a lot of impressive work being done in the Wuhan laboratories along those lines, and that's what I'm most impressed with.
How about you, Angelique? What did you think of the study of the wastewater and how that fits into all of this?
I was, from the very first paper that Monica mentioned, which I also read in sort of like, you know, live feeds almost, because right now we're having this to drink from a fire hydrant of publications. But I remember that one was actually really striking. And I was fascinated by it. So I'm absolutely fascinated by what Monica is doing. And I think it is really something that a method of surveillance that should become standard and routine throughout the world.
Mm-hmm. How about you, Bruce?
So this demonstrates the importance of layered responses to the COVID-19 coronavirus pandemic. So we hear reports of the number of tests, for instance, the number of people who are being tested right now. That's far from enough to help us understand, first of all, what type of surveillance is in place, and also, you know, how the virus is actually spreading. So what we need in our country is a very organized, multi-layer surveillance approach, where it's a combination of testing people, and as well as, you know, looking at serosurveillance, so the people who've already been exposed, along with environmental surveillance, such as what Monica indicated, so that we can combine this data to develop an active dynamic map to really show where the virus is moving. Mm-hmm. And that's been one of the biggest challenges with our response, because otherwise we're left guessing. It's basically like playing a football game without actually seeing the field or without seeing the opponents. That's one of the reasons why a lot of the closings and openings have been very haphazard, because we don't have a line of sight to the virus. We don't know exactly where it's moving. We're relying retrospectively on case reports. And we know that there's the delay, you know, potentially a week, 2 weeks, maybe more weeks before a case actually, so someone actually has to go to the hospital or go to the clinic and be reported as a COVID-19 case, because then they get tested and then we hear about it. And that's, you know, a 2 weeks to a month delay. So we're actually behind the times. Like, we don't even know what the trends are actually occurring right now. We're actually seeing what's happening a few weeks prior. So if we do something like, for instance, wastewater or sewage testing, that gives us a better sense more proactively of what's actually going on. So it's really needed work and really needed research. And I also wanted to point to Angelique's work. This is very important as well. We have to keep in mind that there are a lot of conspiracy theories that are out there that claiming that this is bioengineered, et cetera. there's so many examples of viruses jumping from other animal species to humans. This repeatedly happens over and over again. So we have to keep in mind that this is not going to be the last pandemic. No. And so that to better understand these mechanisms is a whole lot more important than tossing out conspiracy theories to really ascribe blame and things like that. We really need to have a better understanding of the natural evolution of this virus so that we can help deal with this current pandemic, because who knows, there might be mutations or more jumping during the pandemic, and also protect against future pandemics. And I also want to point to Paul's work. That's really important because we need multiple vaccine candidates. We need to have multiple possibilities because our study that was published in the American Journal of Preventive Medicine recently showed that if you want a vaccine to completely eliminate social distancing, it has to be at least 70 to 80% vaccine efficacy. That's a pretty high bar. So most likely what will happen is we'll need multiple vaccine candidates to come out, and then we'll have to decide how to use each of those vaccines in combination or isolation and targeting the different populations. So the more candidates, the better, because each candidate may have its own relative strengths and weaknesses. So it's great to see that work being done. And we also have to keep in mind, we should not rush a vaccine. We cannot rush a vaccine. The vaccine has to be efficacious, and most of all, it has to be safe. It has to be safe to everyone.
So what Bruce was saying about the new pandemics, he's absolutely correct. And I just want to remind everybody that SARS-CoV is not the first pandemic of coronaviruses. It's the third pandemic in 20 years. This is really bad, right? So this means that there might be currently a pandemic brewing. And in terms of Monica's work, it's absolutely essential that her work becomes routine in surveillance.
Yeah, that's a good point. Coronavirus has been a concern for a number of years. I mean, the public has only heard of it recently.
Yeah.
But we're part of an NIH network that basically develops computer models to help respond to any infectious disease threat to the United States. So, for instance, in 2002, 2003, that was the original Sars outbreak that actually evolved and moved to. We saw cases in Canada, as well as in its origins in different parts of Asia. And then there's also MERS, which stands for the Middle Eastern Respiratory Syndrome. And there was a warning 2 years prior to this pandemic on the NIAID website, that's National Institute of Allergy and Infectious Diseases, saying that coronavirus is one of the viruses is high risk for causing a pandemic. Because it's a highly mutable virus. We've already seen multiple different versions of it. And we've also seen cases in which there were outbreaks with severe consequences.
Bruce,
So,
a question for you. At this point, do you see a resurgence, a second wave, and do you see us going back on lockdown?
Yeah, so I think so. In terms of the terminology, we're still within our first wave. In terms of what's happening is the virus was imported into New York City because it was a large population hub with people who are infected in Europe. And then you see this geographic spread. So it started off in New York City, and then it's been spreading progressively throughout the United States. So that's still the first wave. From a geographic standpoint, there's different places that have seen more surges, but the virus is still spreading in the New York City area. So we're not, looking at, for instance, Monica's work, the virus is still present in New York City. So the question is, will we see an upswing in New York City as well as other places? And the concern is yes, because if you look at the natural history of many respiratory viruses, including different types of coronaviruses, they tend to demonstrate a seasonality. And what do I mean by seasonality? That means its transmission, its risk of transmission, its rate of transmission tends to go up and down with the seasons. And so if you, for instance, if you look at the flu virus, people don't tend to get sick with the flu during the summer in the Northern Hemisphere. And one of the theories that was postulated because of some studies that have been done with rabbits is that when the humidity goes down, the virus becomes more transmissible. So respiratory viruses become more transmissible because you have smaller respiratory droplets and they're more concentrated with virus, and it's a higher infectious dose per volume.
Possible lockdown?
Possible lockdown, because we also see schools opening and businesses opening, so the virus might spread more actively. So the only way to really suppress that would be to close businesses and close schools again. So we have to be mentally prepared for that possibility.
Yeah. Well, it's great that CUNY is so involved in the work being done to create a vaccine. There's some very important work going on across the university. I want to thank our guest, Queensborough Community College Associate Professor of Biology, Monica Trujillo, researcher and associate professor at John Jay College, who's been studying bats and COVID-19, Angelique Corthals, professor of health policy and management at the CUNY Graduate School of Public Health and a contributor to Forbes magazine, Bruce Y. Lee, and microbiology professor from the CUNY School of Medicine, who's also a researcher and scientist, Paul Gottlieb. Thank you all for joining us for our roundtable discussion of vaccines and the work being done at CUNY. And thank you all for your work. We really appreciate it. And I want to thank you for joining us as well. I'm Mike Gilliam. Stay safe, and we'll see you next time.